Well, thank you very much, Max, Jan, for inviting me. This is, I think, for the first time where I can speak. I'm a physicist by training, so everyone reflects first who is here. I'm a physicist by training. I was doing interest in theoretical physics, and my first work on biology of aging was in 2015. And I think this is, I'm a happy physicist today because for the first time, like in the world where every inline equation reduces your citation twice, you can actually show some math. So that was a disclaimer. If math offends you, this is not the right talk. So, well, I'm super interested in aging. This is like focus of my research. And of course, everyone has its own definition. I don't know how it works. Does it work? The mic? Yeah. So, is it on? It should be. Can we? Okay, I can try to say louder for the moment. So look, the practical definition of aging, I think, is the exponential acceleration of our chances to die as we age. At least this is the most robust definition that I have heard. And the chart on the right, this is from Kalika paper on naked mole rat. So this is the mortality as a function of age for different species in dimensionless times relative to the development time. And we can see that humans are unhappy creatures because we have just ridiculous range of mortality acceleration compared to everyone else. My feeling is that if anyone wants to extend human life by more than a few years, we should understand what actually changes the slope of this curve. And what I'm trying to present to you today is an attempt of a physicist to actually explain at the end, this is the kind of spoiler, at the end there will be an equation with validation that actually shows you what the mortality rate doubling time depends on. Oops. Okay, the promised math. So we have heard today that, well, biology is complex, there are many parameters, so those X that we measure, that could be DNA methylation, transcriptomics, and everything else. We can measure hundreds of thousands of parameters from every specimen right now. The problem with this is that this number of parameters is actually very small. We don't measure most of the properties of the organism, so this is always a small window on the aging guy. So we have been presented today, but does it work? I don't know.
This one's set better.
Yeah, what if I'm just talking louder? Does it help?
Yes, this one's set better.
Yeah, this is set better, cool. So look, we have been presented today with approaches. Okay, much better now, thank you. Well, aging is a dynamic process, so we have to understand how the changes of physiological parameters depend on the state of the organism. So we have seen today talks when people tried to derive these equations, actually get lots of biological data and try to fit the biological data in the dynamics on these kind of equations. My approach is a bit simpler because obviously, well, there is some kind of a Yong state. The physiological variables change, and you can linearize those complicated equations into some simple form. And the only important quantity here is this matrix that tells you how different physiological variations affect the change of the variations, right? So this is like your connectome. This is the structural property of the regulatory network. Basically, your stability property of the regulatory networks depend on the properties of this matrix, like your connectome. And if you think about this, these are huge matrices. They are so huge that I think never we will know even every component of this matrix. It doesn't make any sense try to actually understand how these individual components behave. What is important is that there was a work in 2008 by Stuart Kaufman, as a biologist who turned to be a physicist. He tried to infer the interactions between different components of regulatory networks in different organisms in 2008 from transcriptomes. And he found that with experimental accuracy, he cannot find out if those species are stable or not, if their regulatory networks are stable or not. So basically, if you look at the spectrum of this matrix, you would know at the end that certain eigenfrequencies, certain time scales that are associated with stress responses are slow, certain are very fast, and the one that is closest to zero, the one that has the slowest dynamics, actually is responsible for stability, instability, or responsible for aging. So basically, our first proposal was that those networks that are unstable are aging organisms, and those networks who are not stable, those networks that are stable are probably non-aging organisms. So when one of these eigenfrequency crosses zero, then the whole dynamics of the system is dominated by just one mode. This is called criticality, meaning that all the physiological changes are driven by one variable. And that's what Uri was talking, I think, about roughly today in the morning. I don't care what is this variable. This variable exists. This is like, in physics terms, this is the order parameter of the stability-instability transition, but you can call it damage. And the dynamics of this damage variable could be for unstable animals, it's like in unstable potential, and for stable animals is like in stable potential. So basically you can have two kinds of situations here. Obviously in unstable animals you will have exponential disintegration, and in stable animals you would have age-independent mortality. So this kind of very simple model explains no aging and aging just by a smooth change of just one variable, which is the eigenfrequency, the smallest eigenfrequency of the connectivity matrix. So, of course, I mean, this is like theory, but in practice, you can identify the parameters of these models from longitudinal biomedical data. And to identify dynamics, you have to have longitudinal data, and that's what we do. We take biomedical data of animals or humans today, and we try to predict this biomedical data in the future. So if these models are set up correctly, these models identify the parameters, and one of these parameters, for instance in mice, works like a biological age. One of the variables in this network is exponential, so this damage is accumulated exponentially. There is a certain level which is incompatible with survival, like we have just been presented in a few other talks. And this variable that is inferred by the network, this order parameter or the damage, actually correlates with hallmarks of aging, like, for example, the number of senescent cells quite better than the chronological age. So this is the true biological age. You can call it damage, you can call it biological age, doesn't matter. This is just one variable that has exponential dynamics, at least in mice. If this works again, I don't know. Yeah, so how do we find out if animals are stable or not? And there is, of course, just a practical definition how to do that. In order to understand if an animal is stable or not, you have to compute what is called an autocorrelation function. It tells you how is an animal similar to itself across lifespan or across a time difference. So in unstable animals, this autocorrelation function should not decay, should be even amplified. And in stable animals, this autocorrelation function should decay as a function of time between the measurements. So this is the actual data in mice. So this is the biological age at a certain age, and this is the biological age blue at 14 weeks ahead and 24 weeks ahead. And you can see that there is quite a perfect correlation. The autocorrelation function in mice doesn't want to decay, which means that mice are precisely unstable animals. And this, by the way, has a very interesting practical consideration. If the autocorrelation function doesn't want to decay, thank you. So if the autocorrelation function doesn't want to decay, it means that the system has memory. Meaning that if you have an anti-aging intervention at some young age, even if the intervention is given in a short time, the effect of the intervention should stay with the system forever. And that's exactly what happens in experiment in mice. This will be published eventually. This is one of the experiments. You have two groups, treated, untreated. You have an anti-aging treatment in between, no treatment later on, and you can see that the treated group never matches the control. So in unstable animals, you can actually achieve rejuvenation. And mice, I think, is a kind of archetypical example of that. There is something terribly wrong in humans if you look at the human data. So first of all, if you look at basically any parameter that you can measure as a function of age in humans, that parameter would not go up exponentially in humans. This is kind of interesting. We have huge mortality acceleration range in humans, right? The mortality is going up very strongly. And at the same time, there are no variables that are changing exponentially with age, which means that aging in humans is a second order effect. We're actually a long-lived species. Now, the most interesting thing is that if you try to measure the autocorrelation function of physiological parameters in humans as a function of time difference, you would find that the autocorrelation function disappears in a few weeks. Remember, in mice, the short-lived species, the autocorrelation function was constant in 14 weeks, 24 weeks. In humans, the autocorrelation function decays in a few weeks. That technically means that humans are stable, dynamically stable animals, very much different from mice. This is an inhuman way to measure the autocorrelation function. This is inverse hospital stay. This is another way to measure resilience. The recovery time, as you can see, the recovery time is going down as we age linearly and hit zero at 110, 120, as Uri suggested. And if you measure the variance of biological ages, and these are different biological ages, one of them is pheno age, for example, you can see that the inverse variance is actually also going down linearly with age and hits zero at about the same age. This basically means that the fluctuations of physiological variables in humans can be mapped onto Langevin equation. This is a stochastic equation that implies recovery. So there is a force that returns to the equilibrium. And the power of this force, the strength of this force, which we call resilience, is actually going down with age linearly. And that's why the variance, which is inverse resilience, diverges hyperbolically, and that's why inverse variance is also going down linearly. So basically that tells you that humans are born stable. This is just a manifestation of that. And our stability, our ability to recover is degraded as we age by another process. And this degradation, loss of resilience, makes us incompatible with life at some maximum lifespan, which is about 110, 150. I mean, it's hard to estimate more precisely. The interesting point here is that all this data is consistent with the idea that the resilience is affected by aging, but the power of the fluctuations is not affected by aging. So essentially, it's a loss of resilience at a constant temperature for those who understand this language. So it's quite, I think it also reflects to the previous talk, because obviously nobody lives up to 120, almost nobody. Actually, the resilience is lost, the stability is lost earlier because of the fluctuations, like in this example in flies. Basically, fluctuations make us leave the stability basin earlier, and we start disintegrating as mice. So basically, we have two kinds of people in every population, stable and unstable, and actually we have a publication where we can prove that. So now, the most interesting question is, of course, what drives those fluctuations and what drives that loss of resilience, right? So what is that? So for that, I will kind of go back to thermodynamics because, well, in some very, non-reductionistic picture, what living systems do is that they generate energy and use it to produce useful work, and useful work in biology is generating molecules, activating pathways, or building systems. And then, of course, no amount of energy can be translated into useful work only. Quite a lot of that energy is dissipated as heat, unuseful energy, which is present in the system and attacks the system, producing damage. So how it works, first metaphorically and second with the data, is like this. So if these are all your kind of molecules, important molecules in your body, like listed in some random order, then some of them work in concert. And these are your pathways, right? These are systems that are getting activated and deactivated in response to stresses. You need energy to activate and deactivate them, and that's what you normally do. And then, of course, the excess energy, the heat, is attacking the system and tries to modify all those molecules in all kinds of random places all the time. So once again, these are coordinated changes and these are uncoordinated changes. This is the difference between the two. These are coordinated reversible and these are just stochastic random changes. So what you would expect is that if you get enough biological data, you should end up in a situation where there should be certain correlated features that have stochastic dynamics, which are just reflecting your life history. And there would be certain uncorrelated features that are just piling up linearly. So the longer you live, the more such features you get. And by the way, this is precisely what you get. For example, if you do principal component analysis on DNA methylation, in humans, you would find one feature that is going up linearly, and its variance also, by the way, is going up linearly, which is kind of a hallmark of independent noise. And then you have at least one feature that is hyperbolic. It diverges at 120, and its variance is also diverging at 120. So this, how do I know if these are correlated, coordinated, and these are not? I mean, Poissonian noise is a nice property, but how to prove directly that these things are uncoordinated? Well, we go to mice. In mice is the same. We'll find one feature that is exponential. Mice are unstable, if you remember. And then there is another principal component that is linear. I mean, principal components don't have sign, so we don't care if it's going down or up. And then we go to single cell methylation data. And in single cell, for every site, if these are methylation sites, we can compute two quantities. We can compute how much this site participates in this exponential feature, and how much this site knows about the changes in all the other sites. And you can see that the sites that are participating in this exponential feature actually have high mutual information relative to the other sites, which means that the exponential feature is indeed the coordinated feature, and the linear feature are the guys who change, who flip independent from each other. By the way, if you have a trillion cells with lots of molecules and you have a bunch of them changing independent from each other, your configurational entropy is going up exponentially, meaning that the flipping of those sites increases the configurational entropy of the system. And by the way, when you hear the word entropy, that should scare you off, because entropy doesn't want to go down normally. So what happens is that when you apply anti-aging interventions, the dynamic modes respond to interventions very well, because they are evolutionarily selected to work, to respond to stresses and go up and down. So this is parabiosis, which we used Vadim Gladishev's data. So this is this dynamic signature. So young animals are younger than the old animals, and animals right after parabiosis are younger than non-treated animals. And by the way, effect stays forever, right? Because these are unstable animals, right? This is rejuvenation. Not so good in long-lived mammals. This is our result. These are dogs. So this is the typical effect, DNA methylation, the correlated feature on a drug that works against aging in dogs. You can see that before the treatment is higher than during the treatment, but once the treatment is off, the effect of the drug disappears. Wow. So I'll try to accelerate right now. So no anti-aging intervention actually works against the entropic features. As you can see, the same parabiosis doesn't change the entropic feature because, well, we all know entropy doesn't want to go down. How do I know that this is what we can learn from this relation to entropy? You can actually try to understand what is the damage accumulation rate, and the damage accumulation rate is actually dependent on the property of the free energy surface. So your life history is a constant sequence of jumps in this very sophisticated surface, and the rate of these jumps depends on the probability of the activation barriers. If you look at the actual loading, if you look at the actual rates of DNA methylation transitions, this is the log probability of the DNA methylation rates, you would find that there is nice exponential feature here, and you will find it everywhere. And this exponential feature actually means that the probability of barriers have this what is called Gumbel distribution. This is the extreme statistic distribution. And that basically tells you that these jumps, that the aging, this irreversible aging, is a collection of jumps, and those jumps are rare events that occur when you simultaneously have failures in multiple redundant systems, which means that in order to revert that, you would have to recreate the same situation, which is hard. And Stephen Wolfram has just published a piece explaining why most of human aging is irreversible in a slightly different language. So I'm now kind of accelerating very quickly. I will not explain that. Basically, I'm telling you that if you try to build a minimalistic model, you would find that it will have two properties of every aging organism. One of them would be the damage accumulated so far, irreversible damage, configurational entropy. And the other would be the temperature. So the temperature is actually a parameter that controls the power of stochastic noise across pathways. And that's what we've done. We identified pathways in DNA methylation data, and we demonstrated that every species has a parameter. This is fluctuation dissipation theorem telling you how variation of pathways is dependent on the resilience. And you would find that this is the temperature that we inferred, and these are the variabilities of pathway activations. You can see that there is indeed one parameter that controls activations of multiple pathways. And to complete this story, the difference between the maximum and the minimum human lifespan, I mean, you can derive, this is analytical model, you can actually derive that the Gompertz exponent should be inversely proportional to the temperature, and the difference between the minimum and the maximum lifespan is actually proportional to the temperature. So the temperature in long-lived mammals is controlling the difference between the minimum and the maximum lifespan. Yeah, I'm like getting out quickly of that. So we were able to show that we can measure this temperature in dogs. You can see that this is mortality is a function of age. You can see that the mortality is indeed inversely proportional to the temperature, and the difference between the maximum and the average lifespan does depend on the effective temperature across mammals. That basically brings me to my last slide. This is aging phenomenology. So see how long we could go without even starting talking about biology, by the way. There was almost nothing there here, nothing there about biology. These are just mathematical constraints that exist on biology. They are kind of supersede biology in many ways. So basically, all animals can be separated into short-lived and long-lived, as it was suggested today, where the average lifespan and the maximum lifespan is on the left, on the right. And hallmarks of aging are in unstable animals. This is my last slide. Are correlated. You hit one, you reduce all the others. They are decorrelated in long-lived species. That's why we have drugs against individual diseases. Drugs against hallmarks of aging, work very well. Aging is mostly reversible in short-lived species, entropic irreversible in long-lived species, and fluctuations, this effective temperature has small effect on lifespan on short-lived species, and will have very strong effect, like difference between maximum and average lifespan is 40 years. So basically that's it. We propose to focus on noise in long-lived species rather than on individual hallmarks, because we believe that can bring the lifespan, increase it by 40 years, maybe. That's very feasible to do. And the ugly truth is that those drugs will not intercept functional decline. So to intercept functional decline and improve maximum human lifespan, we have to do something else.
What's your definition of short-lived versus long-lived species? What is the time separation? How is that determined? Because biological systems are fundamentally there's a huge amount of concentration.
I mean, the formal definition is that loss of resilience occurs before or after the average lifespan. So in short-lived species, like in mice, they are not resilient almost from the start, so their average lifespan is larger than the age of loss of resilience. In humans, the loss of resilience occurs in 120, and the average lifespan is below that. So basically, you can separate all the animals by the age of loss of resilience and the average lifespan. So humans are definitely long-lived according to this definition.
Thank you for the tremendous talk, as usual. My question is very specific, actually, regarding the detail on the slide with two PCA components on DNA methylation. It would be better if you show it, but if it's possible to show.
I will try.
Yes.
It would be possible.
I specifically interested.
Which one?
This one. Yes. The first component is related to the linear change, as you said, and the third component is related to this exponential. What about second component?
Well, I think it's Batch, but I cannot prove it. I think it's a Batch effect because it doesn't enrich any biology. It should not be according to the model, and all this data is dominated by Batch. I mean, we are lucky that we have biology in the first few principal components.
Okay, I have second question, but probably by one, okay?
Hi, thanks for the fantastic talk. It's very illuminating. So my question has to do with the, let's say, the molecular or physiological nature of temperature. So in humans, what's the best guess in terms of what, at least on what level is the temperature, whether it's at the physiological level, cellular level, molecular level?
Well, this is non-equilibrium system, which means that there may not be a global temperature. So obviously on the scale of molecular interactions, it's body temperature, but we correlated the effective temperature from mammalian data set. It is not the body temperature. It's the effective temperature that exists, that controls the fluctuations of slow pathways, I mean, much slower than molecular interactions. So, well, obviously we have ideas. I mean, of course we correlated this temperature to everything, and there are certain pathways that are presumably controlling this temperature. So I hope that within a year, if the validation in experiment happens, we will start, presenting the actual biology here. So we have ideas, but we don't yet have proof. So retrospectively, we have correlations, but we haven't yet established that these pathways do indeed control the temperature.
Thank you.
All right. Yeah, I'm wondering, under your model, shouldn't epigenetic clocks, let's say especially first generation clocks that have a higher entropic component, be better at predicting mortality outcomes than second generation clocks? What am I missing here?
Yeah, you're talking about first clocks and Grim age-like clocks. Yeah, I think this is correct. I mean, we have two principal components there, which means that these clocks in some random combination, they pick one or the other. Yes, the first generation clocks have better. The problem that if they are produced correctly, they are not actionable, right? I mean, really, the clock that has the entropic part only has better prediction of mortality, but unfortunately, it's not actionable. And even the first generation clock has sufficient admixture of the reversible component.
Thank you for your presentation. I have one question about the last slide. When you compare short-lived species and long-lived species. And the question is that for the reasons of aging for short-lived species, he's written that it's reversible. But I don't really understand how aging can be reversible, as aging seems to be an irreversible process. So if you age, you travel through an irreversible process if you age. So I wanted to just check this.
Well, the key word here is mostly, because if you look at my slides, I can detect the reversible component of aging even in mice, right? So mice shows both reversible and irreversible component. The irreversible component is linear, and reversible component is exponential in mice. And when you have exponential problem, you don't care about the linear problem. So fortunately, the exponential problem is reversible. If somebody would find a way how to control the exponential component in mice, make mice into flies or humans or dogs, then of course their maximum lifespan will be defined by the reversible component. I'm just saying that relative to, right? So the exponent is so fast in mice, then the irreversible component doesn't have a chance to contribute to lifespan.
Mm-hmm. So that means that in the default settings, in mice, reversible component dominates the irreversible?
Yes.
Because of the order?
Because it's exponent. When you have an exponent, it dominates everything.
But they age and die.
Sorry?
But they age and die. So mice, they age and die.
Yeah, they die. But they die because they have this aberrant pathway that is going out of control exponentially and kills them.
Okay.
So it's a model. I mean, it's a very simple way to die. So they literally die from certain metabolites, whatever, certain factors going up through the roof. That's what was discussed today many times.
Okay, got it. Thank you.
Okay, always enjoyable talks. Thank you. I wonder if it's really warranted to separate so strictly reversible kind of entropic damage and dynamic, because much of the damage is conditionally reversible. So, for example, in the case of heterochronic parabiosis, when you connect the mice, some of the damage may be exchanged, even if it's reversible, like in the old mouse. So, as a result, it could be really, I think it's much more complex kind of.
Well, Jose, I'm always trying to use words mostly and approximately. I think these two things have different dynamic properties, so that's why it's worth to separate them.
I just wonder if it's continuum from one to another, not just two.
Well, even if principal component analysis is able to separate the aging signatures into things that respond to parabiosis and do not respond for parabiosis, I think it's sufficient reason to keep them separate. And also, if you look at this rate distribution, this appearance of Gumbel distribution, it doesn't show up in the reversible component. So that means that they are very different.
Do we have time for one small question?
Who controls the time?
Regarding the slide previous to the last.
Sorry?
Regarding the slide previous to the last one. Yeah, could you show? Yeah, the slide with effective temperature. Yes, I noticed that the p-value here is quite large, but probably this new result.
Yeah, I think the new result is better. So
this new result is only for dogs.
Yes.
And the right part of the slide.
For all mammals. So I believe that since there is a lot of variability between the species, it's harder to see. But if you start looking at one species, also for humans, if you measure temperature in humans, there is also very nice picture with slopes.
But is it expectable result that effective temperature is poorly correlated with the relation between life is maximum?
So the lower is the temperature, the faster is the actuarial aging. I mean, the damage accumulation rate does not depend on temperature, but the perceived aging, actuarial aging from the survival curve is steeper. That's what, by the way, social status is doing.
Sorry, that for mammals, for a lot of different mammals, the right.
The right is for different mammals, so that's why there are structural properties of the networks that are different, and temperature is different. So that's why I believe it's harder to see the effect of temperature only. If you go to one species, then the structural properties are the same, and only the temperature is different. So that's why it's much easier to see the effect.
Thank you very much.
And it's really, we never met, but the ideas are so similar. So this whole, amazing to get together. About the entropy, biology is amazing at lowering entropy, like us. If entropy means we'll be spirochondrium. So if you take an egg from a 40-year-old woman and a sperm from a 40-year-old man and put them together, you get a 0-year-old baby.
Mm-hmm.
So there's some way to actually erase the entropic part.
Yes.
In principle.
Yeah, I think all known examples require to reduce trillion cells to 1, order of 1 at this time. So it's like restoring from backup, right? So if your computer is too messy, you just go and restore from factory backup. It's a new computer, but you lost a little bit.
Thanks, the speaker again.